Hello and welcome to our Global Thrombosis Research Seminars, Network to Network. My name is Stavros Konstantinides from the University of Mainz in Germany. I'm currently the chair of the Invent VTE network, and I'm very happy to host today our webinar with Professor Eric Vicaut, whom I will introduce to you within a few seconds. So what is the Invent VTE network? It's a partnership of 11 national VTE and thrombosis in general research networks. It supports and promotes international collaboration between networks to produce relevant, high-quality academic VTE research. The Invent VTE network of networks is meanwhile present in many countries around the world, not just in one or two continents. You can just see here networks from Australia, New Zealand, Canada, China, France, Germany, Ireland, Italy, the Netherlands, Norway, the United Kingdom, and the United States. Our seminar series are limited webinar series, so they are quarterly seminars. These are not general educational events or webinars on thrombosis, but they do feature select topics, inspiring and impactful international thrombosis research topics and initiatives, collaborations, and efforts from across Invent network member countries. So it has to be something which is relevant, impactful, and also innovative, as you will just hear in a few minutes. You can have access to past seminar recordings. You can see the link on this slide. And we already wish to thank Sanofi for sponsoring this seminar with an unrestricted educational grant. Today's presentation has the title Adaptive Trial Design, Advantages and Drawbacks of the Bayesian Approaches in Clinical Trials. And we're featuring Professor Eric Vicaut from Paris, France. I will say a very few words about his CV in a few seconds. Professor Vicaut, who is a very international well-known biostatistician and MD, medical doctor, and also a very good friend for almost 25 years. And the learning objectives of his webinar would be to present the principles of the Bayesian statistics applied to clinical trials, to illustrate the advantages of Bayesian approach in order to increase the feasibility of adaptive designs, to report the discussion about possible drawbacks of Bayesian approach in comparison with the frequentist approach, that's the traditional classical approach we all know, and to discuss the potential impact of the Bayesian approach on the future of evidence-based medicine. So before speaking about Professor Vicaut himself, I would like to say that we will have a 15-minute Q&A session at the end of the webinar. Please let us know, please then speak up and let us ask your questions to Eric Vicaut or any comments you may have, or you can also write them on the chat and I'll be happy to read them out for you, whatever you prefer. Professor Vicaut has both a medical degree and a PhD in biostatistics and applied mathematics. He was recruited at INSERM as a research fellow and actually became research director of the INSERM in France in 1992 already. And he became professor of biophysics at the Medical University Paris Diderot in 1997. Professor Vicaut is the head of the clinical research unit, Lariboisière, Saint-Louis, and the Center for Medical Devices Evaluation. And he was co-head of the National Platform for Clinical Research, the FCRIN partners, APHP. He's a methodologist and biostatistician, and has been involved in a very large number of multi-center international clinical trials with co-authoring more than 650 articles. And he is also a methodological expert for many government organizations. So the French Market Approval Committee for Drugs, the French regulatory agency, the ANSM, Agence Nationale de Sécurité du Médicament, and also for the French Committee for the Medical Devices in the Haute Autorité de Santé. He has been president of the working group Medical Devices for the Assise du Médicament and the author of the ministerial report Dispositifs médicaux pour une évaluation à la hauteur des enjeux. So Professor Vicaut is a scientific expert, and he's also an expert that has he exerts a very important counseling function for the French government. Eric, we very much look forward to this exciting topic. I know that you're an expert. We have had many discussions on this topic, the two of us and also with other colleagues. So very much look forward to what you will tell us today.
Thank you very much. I hope everything is okay on a technical point of view. I'm very happy and grateful for this invitation. I just will share my screen. And here, I'm just waiting.
Yes, we can see it very well.
Okay, so that's perfect. And I will speak here about Bayesian statistics. But obviously, it's not a talk for biostatisticians. It's a talk for clinical investigators, medical doctors involved in clinical trials. And first, I just want to say a few words about the two big paradigms for biomedical statistics. One is, I will say now, the classical one, the frequentist, and the other one is the Bayesian. But on a historical point of view, even if now we can say that maybe the Bayesian statistics is the modern choice rather than the frequentist one, at the beginning, statistics were Bayesian. And frequentist approach was chosen, I will say, in the early years of the last century for theoretical reason, especially discussed by the Greek statistician Fisher, and also for operational reason. And operational reason is that at one point of the calculation, Bayesian statistics will require a large power for calculation. And it was evident that with, I will say, without computer and even without the modern computer, it was not possible to make statistics with the Bayesian approach for samples that are not very small samples. So one part of the Bayesian statistic has been developed in the 60s for very small samples. And with the new computers, the most powered computer that we have now, it became more and more feasible to address the question with Bayesian statistics. But at this time, we have to say that if you see the papers in medical journals, most trials are made with the frequentist paradigm, and most regulatory authorities' advices and discussion are based on the frequentist paradigm. But my personal opinion is that times are changing, and we will see the landscape of statistics. We will see this change in the few years. I will try to show you in a way as simple as possible what are the difference of the principle of Bayesian statistics. I would say first, the Bayesian statistics is a statistical approach that can use prior knowledge and observed data and combine it to make inferences about the probability of an hypothesis. So it's very important to consider and to understand these basic principles. And this basic principles is quite opposite, quite different than the frequentist statistics. That the frequentist statistics considers the probability of observed data given an hypothesis. And I will just mention this here. I will say, frequentist statistics focuses on the probability of observing a set of data given on a hypothesis. So we can say very simply that frequentist idea is to consider the probability of the effect knowing the cause. So we will test the idea that the observed data are more or less compatible with an hypothesis, or if it's not, we can reject the hypothesis. And we go in the evidence medicine by rejecting hypotheses. And this is the basis of evidence-based medicine with the frequentist approach. The Bayesian approach is, I will say, the symmetric in some way. We try to calculate the probability of hypothesis given the data, and not probability of the data given an hypothesis. So it's the reason why I say it's symmetrical. So we can simply say that the Bayesian paradigm try to calculate the probability of causes knowing the effects, and not the effects knowing the cause. So once more, it's very important to see that the basis of the two approaches are completely different. And it's very important because I will, after my talk, conclude on the fact that it's not only a question of techniques, it's a question of philosophy. And it's very important to consider the Bayesian approach not only as I would say, a technical advance based on the same frequencies. We have to change our way of thinking. Otherwise, we cannot understand the things well, and we can make some errors of interpretation at least. The important aspect is that Bayesian statistics not only consider the observed data of your study, but can incorporate in your way of thinking what are the knowledge before your study. So it's important to consider that that can be an enrichment of your study based on the prior knowledge. This is a possibility. In some way, it's not mandatory, because you can say also using Bayesian statistics, but you do not know something. And you can incorporate your absence of knowledge in Bayesian statistics also. What are the, I will say, the three schematic steps in Bayesian statistics? So step one is to consider what you know before your study. So it's very rare that your study is the first, I will say, the first study in a field. So you can consider in some way that you can have an opinion based on previous study, either using, I would say, some kind of subjective opinion, or for instance, based on meta-analysis, for instance, that you can have made before you study. And this is the first step of the Bayesian statistic. What do you know before you study? That's the first step. But once more, if you do not know nothing, if you do not know anything, okay, say, I have no knowledge. That's not a big issue. So the other step is really your study and your observed data in your study. That's the second step. And the first step is the fact that you can combine what was your prior knowledge of the field with what you have observed in your study in order to have something that combines the two aspects of the opinion and say, okay, I had an opinion before, I have made my own study, I have an opinion after, and in some way that combine my prior opinion to what I observe. The prior opinion can be neutral. The prior opinion can be enthusiastic a priori, but the prior opinion can be very skeptical. Okay, you can say, I'm very skeptical, but I can't change my opinion with data. The only thing is that Bayesian statistics allow you to do this in a formal way. And each step of the Bayesian statistics is related to a statistical function that we will describe very simply here. The first step is known as the priors. The priors are the statistical distribution that correspond to your opinion or knowledge before you study. And for instance, I have illustrated the notion of prior with this scheme. And if you see this scheme, this figure, you can see, for instance, the prior knowledge about a treatment and, for instance, the effect of treatment on mortality. And well, for mortality, for instance, in order to formalize the treatment effect, you can formalize the treatment effect by relative risk of mortality. And if the treatment has no effect, the relative risk of mortality is 1. So that doesn't change. But you can say, okay, I have no knowledge about this. If you don't have knowledge about this, you will have a prior that is like the blue one. Okay, you see the arrow here. That means that I have no opinion. I have no opinion. That means that the risk, the relative risk can be anywhere with the same probability. That is known as a minimally or non-informative prior. But I can have also other opinion. For instance, you see here, this is a prior related to a very enthusiastic prior knowledge. What does that mean? That means that if you are enthusiastic, the prior say, okay, I think that the highest probability of the relative risk will be lower and largely lower than 1. This is very enthusiastic. At the opposite, you can be very skeptical. If you are skeptical, you will say, okay, I think a priori that the relative risk will be larger than 1. I think that the effect is deleterious. So this means that you will have, for instance, a skeptical prior that is like this with a value of high probability for relative risk at 1 or larger even than 1. So you see that you can translate a knowledge or an opinion in a way of statistical distribution showing the probability that translates your opinion in probability of a treatment effect. So that's the first function related to the step 1 of the Bayesian statistics. Then I will do my trial and I will collect the data. And when I will collect the data, I will produce another statistical distribution that is the likelihood function. And the likelihood function expresses the plausibility of a parameter in the light of observed data. So what does that mean? That means, for instance, here, that this is a plausibility of a parameter, you see here, relative risk. So you see that in this case, the observed data, I say, well, okay, the maximal value of the likelihood function is here. So relative risk has a high probability to be at this value. So that is the translation in terms of statistical distribution of what you have observed during the trial. Then the step 3. The step 3 will combine what you saw before the study. That's a prior. And what you have observed during the study, this is a likelihood function in a probability function or distribution that is a posterior distribution probability. So what happened in the example that in the figure that I show here? You had a prior opinion that was really enthusiastic, because here you really imagine that the relative risk was very low. But this is what you have observed in some way during the trial. What is the result of the trial in a Bayesian statistics as a combination of what you have observed and what you have considered before the study. And this is a posterior probability distribution. That's here. Something that is a combination between before and during. Obviously, the more data you collect during your trial, the less important is the aspect that was your opinion before your trial. So you have also this idea that you can incorporate in your results what you have considered before or also have considered before, but in some way, the key point are the result of your data collected during your trial. So the weight of before and during obviously is dependent on the amount of data that you collect. So the result of a Bayesian statistics is always this posterior probability distribution. And it's very important to understand that this curve is your result. Just one thing that we have to keep in mind, that the Bayesian framework aims to define the probability of a desired treatment effect, rather to rule out the absence of any treatment effect. That is the basis of the frequentist approach. So here, everything that we can do, and we will do, is to define the probability of treatment effect. And in order to do this, we will build the posterior probability distribution function, and we will consider it in order to consider our results. And I just want to mention that we have, as a result, we have a function, we have a figure, we have a distribution of probability. But considering this result, we can have many information. For instance, when I have, for instance, this curve considered as, for instance, a posterior probability distribution, here, what can I say with this? Okay, I can say that considering this curve, here, for instance, the probability that the value is lower than this is the area under the curve from minus a large value to this value. And this is the probability that the statistical parameter is lower than this. If I consider another thing, for instance, the probability that the statistical parameter is lower than this value, this is the area under this curve. So I always can consider two aspects. What is the probability that the result is lower than a given value? And I will report my results in a Bayesian analysis always like this. What are the probabilities that the treatment effects are lower or larger than a given value? This is the basis to report Bayesian statistics. So once more, Bayesian's framework aims to define probability of a desired treatment effect. The consequence of this is that we can use all data collected at each time of the study. We don't have to consider the study only when the study is finished. We can consider information collected during the trial, and for many reasons that are not possible in the frequentist paradigm, we can use this information to change also the design of the trial. And this is the basis for adaptive or flexible design. And why is it possible? Because at any time in a Bayesian trial, we can calculate the probability of something in the trial that can interest us and be the basis for a decision in the trial. And for instance, we can use this adaptive function in order to stop early the study in a very, very flexible way. We can analyze every 10 patients and see if we have to stop the study for efficacy, for futility. But we can also very easily stop one arm of the study. And this is very important and useful. For instance, if you have several arms in the study considering several doses, for instance, or several treatments, or several treatment combinations, and you have the possibility at each time during the study to calculate the probability that the treatment effect is, I will say, lower or higher than a given interesting threshold. So that allow you to consider at each time of the study the data collected and use them in order maybe to early stop one arm. I will give some example of this after. On the same, I will say, on the same way, you can also use the same kind of calculation to stop one or several subgroups. And for instance, you can consider a treatment, but you can consider a priori that you have different subgroup of interest. For instance, you can have people, young people, old people, people with large weight or a very small weight. So you can consider all the subgroups, and you can calculate for each subgroup the probability of the treatment effect. And you can consider or the probability of toxicity, for instance. And you can consider, for instance, that for some subgroups, toxicity is unacceptable, for instance, and you will stop this subgroup. And you can do this in a very much more flexible way that with the frequentist approach. That in some way it's possible also to make adaptation in the frequentist approach, but it's more complicated. You can also consider the possibility to say, okay, in order to have a sufficient probability to demonstrate a treatment effect, we have to increase the sample size of the study. And you can have also what is called adaptive randomization. I will show you something after. The principle of adaptation are always very simple. You just have to consider a priori what will be the criteria for adaptation. For instance, we will stop, for instance, the study of a group of an arm if the success of treatment is too low, or if the toxicity of the treatment is too high. And once more, what is very easy is that with Bayesian statistics, you calculate exactly this. what is the probability that the success is, for instance, larger than 20% or 30%, or that the toxicity is larger than 50%, for instance. So you will have directly the answer in order to say you, okay, this is not acceptable considering the criteria that are defined, obviously, prior the study. This is a very heavy slide, but I just have copied something that I have written in a protocol for a new device for radiotherapy. And it's like a phase 2. So this is a very, very innovative device for radiotherapy. And we have made a study in order to have this, to evaluate this very innovative radiotherapy device. And we have made in the protocol an internal control that is standard radiotherapy. But we were very concerned about the fact that this innovative device might be toxic. And then we introduce a Bayesian criteria in order to stop very early the study if we have some information that tell us that the probability of harm is or toxicity is unacceptable. So this is the way that we express such criterion for adaptation. In this case, we say, okay, if we have a toxicity, here it's dermatitis grade 3 and 4, larger than 10%, with an high probability, here 0.9, so that's 90% probability, we can say. Okay, so we will stop. So once more, the way to think about it is very intuitive. We will stop if the probability that we have an acceptable toxicity is high, and we will stop like this. And we can do this for other things. So we can consider also the difference in toxicity between the two arms. And if the difference in toxicity is too large, we can say that we will stop. Or we can consider also the efficacy and consider that is the probability that the efficacy is larger than a threshold value. We can stop the study for efficacy. I have another example here. So it's a 5-arm study. In dermatology, it's also a phase 2. We have 5 arms. But the question is that I would say another way. We do not want to expose patients to treatment with high toxicity or low efficacy. We want to stop this as early as possible. And using Bayesian statistics, we can calculate this. We will make multiple looks of the data. And we say, okay, if we have a high probability that the treatment is good, okay, we will stop for success. We will stop for efficacy. And if we have a very high probability that the treatment effect is unacceptable, we will stop for futility. And once more, what gives us the Bayesian analysis is a calculation of these probabilities that we will use in order to make our decision. And once more, I want to stress that the way to think about Bayesian statistics is that Bayesian statistics give you probability, and then using this probability, you make your own decision based on what you think acceptable or not acceptable, considering obviously your medical knowledge and the other possibility that can be considered. This kind of rules can be applied to other criteria. For instance, you can say, okay, I will stop my study if I have no power to demonstrate anything, or I have to increase the number of patients in order to have sufficient power to demonstrate something. And as mentioned, you can stop some subgroups before. We have also, I will say, something that is, I will say, the end of the process of adaptation, that is adaptative randomization. At each look of the data, you can calculate the probability of success in each arm. And then you will adapt the probability for a patient to be in one arm in function of the probability of treatment effective in this arm. So you will randomize more patients in the arms that are the most, I would say, potentially interesting for the patients, and less in the arm that are less interesting for the patients. So that can be something that can be used in order to avoid too many patients in arms with high toxicity or in arms with low efficacy. So I will try to do in more or less five or 10 minutes an illustration of what is the difference, not only technical, but also, I will say, philosophical in some way, for the difference between Bayesian and frequentist. And that means that we have to think about the basis for changing clinical practice. And we have also always to think about the consequence of the change as a consequence of error when we are changing, but also the consequence when we do not change. This is very important also to consider. I will illustrate this considering, I will say, the possibility to analyze a randomized clinical trial using the frequentist versus a Bayesian approach. And I will use this paper that has been published some years ago in the New England Journal of Medicine, testing the possibility to use ECMO for severe acute respiratory distress syndrome. And, well, I will not go into details, but here, these are the results, and we don't have the significance in this study. And we have all this value, that means that the relative risk of death was observed was this one, 0.76. But as you see, the confidence interval does not allow to consider that it's a significant difference. And the conclusion that it was obviously the ECMO does not allow to lower significantly the risk of death in this patient. And here, Gollinger et al. Has published a post-hoc analysis using a Bayesian approach. And it's very interesting to consider the way to think about this using frequentist or Bayesian. So first, I have to say that in the initial approach, the trial, that was a frequentist trial, was designed to detect, to have the power to detect an absolute risk reduction of 20%. That means a relative risk reduction of 0.67. So it was very optimistic. And it was very optimistic. They have included patients, and they did not, they were not allowed to conclude of a positive effect of ECMO. So the group of Gollinger has used Bayesian analysis. So they have considered different priors Okay, they have used the data of the trial to calculate the likelihood function, and they calculate probability, posterior probability distribution with the different priors. I will not go into the details of this, but I will just summarize some important aspects. So, the Bayesian approach is not to say, can we rule out the effect of ECMO? Is a different question. Once more, it's a different question. It's what is the probability that the treatment, here ECMO, is associated to a clinically meaningful benefit? Obviously, benefit can be considered as relative risk, absolute risk reduction, and we can consider the probabilities of different levels of mortality benefits by Bayesian modeling. And just to say that if we consider just the probability that a relative risk is not 0.67, but something between 1 and 0.67, it's not too bad. And just to illustrate this, for instance, an absolute risk reduction at 2%, just 2%, that means that it's 500 lives saved in US. So that indicates that it's something that can be obviously interesting to calculate the probability that the treatment allows this or not. So we will consider first that we don't have any prior, we don't have any prior knowledge about this. Okay, we say we do not know anything about this. I have no opinion about this. And I will use the Bayesian statistics. And with the Bayesian statistics, I can calculate the posterior probability distribution. And as I mentioned before, I can say, okay, I can calculate, for instance, the probability that the risk, relative risk, is less than 1. And this probability is 96%. So there is a 96% probability to have a relative risk less than 1. So that is to have a benefit, even a minimal benefit, but a benefit. Obviously, the possibility, the posterior probability to have a very large effect is very small here, 18%. For instance, if I consider the information not based on relative risk, but absolute risk reduction, the probability of an absolute risk reduction of 2% or more was 92%. So that means that the result is that the probability that you can save at least 500 patients in US is more than 92%. That is the result of the same data. Just the analysis is different. And the way to think is different. Just we can also make some other or sensitivity analysis considering what we know, for instance, before, because in this case, we had meta-analysis in order to have priors that consider this information in our calculation. And if we do this, we can see that the results are even better because the prior studies there was a trend for ECMO, but it was not sufficient to consider that it was useful. So in order to finish, I will say that what are the difficulties for Bayesian approach? So the first thing is that it's complete for many aspects. It's in contradiction that we have learned or in what I've taught for many years to my students. That's something that is really, really difficult. And it's not contradictory. It's contradictory if you just consider Bayesian approach using frequentist concept. But if you are completely in the Bayesian paradigm, it's not a problem. But there are also technical problems. To anticipate sample size, it's much more difficult because you don't have calculations. You have to make simulations that take time and some degree of difficulty. You have also technical difficulties of clinical trials, for instance, for drug supply, because if you don't know exactly what will be the number of patients in one arm or in another arm, if you have to stop arm, it's a logistical difficulty. The other difficulty is that it requires a close monitoring, because with Bayesian approach, you have to make very important decisions based on what happened during the trial. So it's very important to be sure of the results at the moment you make this interim looks. Another question is the choice of priors that appear to introduce something that is subjective and that can be discussed. But you can base your priors, for instance, in some case, on meta-analytic approach. That is not, I would say, a good way to do this. And you can use always non-informative priors. So you don't introduce something, you don't introduce an a priori knowledge about this. And you can also test the results with different priors, that is Bayesian sensitivity analysis. So the subjectivity of conclusion, but the subjectivity of conclusion also appears in frequentist approach. For instance, in this case of Eolia, you have the conclusion of the paper, but you have here the editorial. And you see that, okay, the paper was negative, but the editorial say, okay, he has some benefit. But with the Bayesian statistics, you can consider this in a formal way. On a quantitative basis, we have quantified what will be the probability of benefit and the probability of benefit depending on the amplitude of the benefit. The subjectivity of conclusion is a problem for regulatory agencies, but Bayesian approach can be accepted based on some very rigid values and the use of non-informative priors. So it's no more a large difficulty. So my final conclusion is that I think that Bayesian statistics can be used, I will say, as benefit to be used, and will change our way to consider what is evidence-based medicine. In some way, it will be much more difficult for us, because the interest of the pragmatist approach is that you have just a black and white conclusion. And it's always easier to have a black and white conclusion, but to say that, well, the probability that if I change, I have a benefit is something. So what will I do now? But I would say this is much closer to what is the medical way to think, rather than, I would say, the hypothetical hypothesis deductive way to think that is much more related to physics or other kind of science. So I finish now, and I will be happy to answer to some questions.
Thank you very much, Eric. That was really, I think, a brilliant presentation. Very difficult topic because, as you said, it's not just a technical issue, it's also a philosophical issue, and there's nothing more difficult than people having to change their minds, right? I mean, this is something which is, but I think you very nicely combined the technical part of it, which is of course very complex and most of us would not understand, but you made it in a simple way, and the philosophical part where everybody should be able to think a little bit and see what may change. I would like to ask all of you or remind all of you if there are, to ask your questions in the chat. And I think may I ask, I have a couple of questions. We have discussed it also in the past. And of course, you know, when you confront physicians with some totally new way of thinking, it's always a little bit difficult. So you said that in that case, you do not have one interim analysis or two, you constantly reevaluate the data, right? And you calculate probabilities. Is that correct?
Yeah,
And
I will say that this can be adapted in some way to different trials.
yes,
Obviously, we will not do this, I will say, every 2 patients or every 5 patients.
okay,
Hence, in some cases, we can do this. For instance, in rare diseases, for instance, I see
yes,
a comment about a rare disease. I think that in rare disease, it's important because obviously in this case, you will consider the calculation of probabilities for only few patients, every few patients.
yes.
So it will be interesting.
Because my question was going to be, when you have an adaptive design or based on the classical frequentist approach. I mean, let's take an example of a thrombolysis study in PE, one of which you were co-author with co-authors together. You say, okay, we have planned to include 1,000 patients, and at 500 patients, we do an interim analysis, and then we decide whether of course, in that case, of whether we can continue or not. Could it be a problem if we just say we will do with the Bayesian approach, we will do more frequent analysis, that we can say, oh, okay, after 200 patients, for example, when we're calculating, oh, now it looks good, now we stop. And if we continued, maybe the probability would become lower again. So would it be do you think it would not be necessary to identify a little bit, you have to have a certain number of patients already. You cannot, you said, of course, rare diseases are a very big problem, but let's take a disease like pulmonary embolism, which is not very frequent like myocardial infarction, but it's not rare. And how do you resolve this? I mean, you say, okay, let's calculate, or are you afraid that people can tell you, now you found the sweet spot, now you stopped, right? I mean, you.
So I will say that you have to consider maybe 2 aspects. The first one is that the more patients you have in the Bayesian trial, the less important are the priors. So
Yes,
you have to consider also this aspect. If you introduce priors, and even more, if you introduce priors that are not non-informative or weak informative, if you introduce this kind of priors, you can say, okay, I want to have a sufficient number of data in order to be in some way less dependent of my prior knowledge. So that's
yes.
one. The other aspect that you mentioned is that, okay, you can have a value in some way, and that after it can change. That's true. But
Yeah.
what is very, very easy in Bayesian statistics is that you can consider criteria to stop that change for the first interim analysis, the second, the third.
Yes.
So you can be more strict for the
Mm-hmm.
first and less for the other. That's also the case in some frequentist alpha value distribution of frontiers in order to when we make interim analysis. But you can do very easily like this. The other, I think that the other thing that is very interesting in Bayesian is that you can combine some rules regarding efficacy and toxicity. Very easy.
Yes. Yes, we have actually some very nice comments also from our participants, whom I thank very much for entering their questions on the chat. There are several very good comments, and some of them were partly addressed, as you said. So what could be the advantages and pitfalls for rare diseases? Now there is here. There is a comment by a colleague, maybe just a few brief answers to try to address some of them. A comment saying that we should perhaps avoid the term early stopping in the Bayesian approach, because early stopping always has a negative connotation, a meeting, and is a frequentist right from the frequentist framework bias, maybe. So people are more skeptic. Should one say, how would we? How should we say early stopping, or what? How should one call this?
I like this. I like the comment very much. It's appropriate stopping. Yes, I agree.
Appropriate stopping. That's good. Yes.
I just want
there is
something that's the question is that it's appropriate stopping if your criterion is appropriate. So
yes.
This is a question of the subjectivity. So you say, in some way, if I have already calculate a high probability that an effect, a treatment effect is so high, I can stop. This is appropriate. It's not,
yes.
I totally agree that it's not early, it's appropriate.
It's not early, yes.
I like the comment very much.
There are also two questions by colleagues regarding sample size calculation. It has not become very clear. Does the Bayesian approach, because I mean, you know, the most important thing that we have to do when we submit an application is to make a sample size calculation. So does the Bayesian approach, how does it, so I understand now you run simulations, so you do not have, right? This is the way you try to calculate the sample size. Maybe you tell us in, let's say, 1 minute, so that we have a couple of other questions as well. So how does now the Bayesian approach affect your sample size calculation?
So
the first one.
I would say that basically we don't really know the sample size, I would say, with a calculation. What we know is that, for instance, we can consider some probabilities for difference between the two groups. Yes. And what we will do is that we will simulate, for instance, 5,000 studies and see with different, for instance, with different sample size, and we will see when the study will stop, considering the criterion that is appropriate for stop. So yes. We say, for instance, okay, I cannot say that my sample size is, for instance, 100 patients per group, but I can say that with different hypotheses regarding the treatment effect, the median value of the trial will be, for instance, 90 patients per group. And so that's the way here is okay. People, if you consider two groups, but for instance, if you consider 4 groups, 4 arms, you have to make simulation considering that you have one arm very different than the other, 2 arms that are similar, and some question about the different, what is called different scenario about this. And you make this simulation and you consider what is something reasonable. And you can say that, okay, with this sample size, I will have a high probability to demonstrate what I want to demonstrate.
I see.
But the other thing that we do also is to consider also a maximal sample size. That is we cannot say that we will go to.
The question is, as a renowned colleague writes in the comments, so what is better for you in order to get the funding for the grant, right? Because I mean, somehow, what the maximal or the minimal size?
Yeah, so I can say what I do. And
yes,
I've got some grants doing this, so I can give this as a tip. So just to say that usually I make the calculation based on the frequentist approach and consider this as
okay, yes.
And then I introduce this maximal sample size in my simulation, and I see what happened. And in most of cases, I calculate what will be the median value of my trial that will be smaller than the calculation by the frequentist approach. And it can be shown that Bayesian trials for the same hypothesis and more or less the same way to think about what is the appropriate threshold for success are require less patients, 1/3
Yes.
less patients than the frequentist approach. So
thank you.
I use the maximum sample size. I make my budget maybe on maximum sample size or not. I can make my budget on it depends, but I can make my budget on the results of the simulation for the median value of the trial. And well, and obviously it depends on the possibilities that I will have to get more funding if I need.
Yes. I think we have a very many further very nice questions, but I mean, we are at 1 minute past the hour. Maybe if I can say a last comment, perhaps for younger colleagues who are watching us, before saying that, I mean, everyone is praising the nice, beautiful way how you explained this. You mentioned the, so let's say a medical comment from my side, or medical statistical. You mentioned the example of the Eolia size, and you said with the frequent approach, this is a negative study. And then you say there is a 92% with a Bayesian approach that the absolute risk reduction, right, was 2% or more. Of course, what even the Bayesian approach does not tell us is how medically relevant is an absolute risk reduction of 2%, right? I think this is something that then must be judged by the medical community, or the regulatory authorities, right?
That's true, but it's in this case, it's not completely true, because in this case, the criterion is mortality. So,
okay, for mortality, but it's an expensive new method, for example, something that you're doing.
Sure.
ECMO is not cheap here.
Yes, certainly. So we have to consider also the drawbacks of the toxicity, of the possibility,
yes,
safety of what we discuss about. But okay.
But this is, of course, but I think this is also what you have told us many times in your seminars. It is not just a statistical calculation. It is also that there must be the medical thinking. There is a steering committee, a scientific committee, and of course, these are people who must also, based on clinical expertise, right, judge the data. It's not just, but so thank you very much again, Eric. I am really very enthusiastic about the number and of the comments, and how happy they are with your presentation, and how they congratulate you. We thank, of course, our sponsors, and we thank all our colleagues who watched us and contributed to this discussion. I am sorry, I think we addressed some of your comments. We can never address all of your comments, but I hope that at least this was very informative for you all, and many thanks to the Invent VTE network for making this possible, and to Professor Vicaut, who could share his expertise with us in such a lively manner. Thank you. Thank you.
Thank you, Stavros. Thank you to the audience.